Transbilayer movement of lipid molecules involves in several enzymes that can catalyze the translocation of phospholipids, sphingolipids and other membrane lipids from one leaflet to another leaflet. And in this process, ATP is needed for this particular translocation. Hello friends, welcome to Biology Concern. I am Animesh, and in this video I am going to discuss about transbilayer movement of lipids molecule. So let's jump into the topic. First one, if we observe the definition, that is the transbilayer movement requires that a polar or charged head group, that means if we have two leaflets are there, so if this is the outer leaflet and if this is the inner leaflet, okay, so that is the requires the polar or charged head group. So this is the polar or charged head group, charged. So in this process, this phosphate head group leaves its aqueous environment and moves into the hydrophobic lipid environment and finally reached into this inner leaflet portion. So into the hydrophobic interior bilayer, a process with large positive free energy chains that requires ATP. So this is the definition of transbilayer movements of lipids. So one is the most common example. So one is the most common example is in endoplasmic reticulum. In endoplasmic reticulum, membrane glycerol phospholipids and sphingolipids are synthesized in cytosolic leaflet and luminal leaflet. So membrane glycerol phospholipids synthesized in the cytosolic leaflet of the ER, endoplasmic reticulum, and sphingolipids are synthesized in the luminal leaflet of the endoplasmic reticulum. So basically these glycerol phospholipids or glycerol phospholipid that is synthesized in the cytosolic leaflet needs to go into the inner leaflet or luminal leaflet, and the sphingolipids that is synthesized in the luminal leaflet comes to the outer cytosolic leaflet. So this movement is catalyzed by the enzymes. Those enzymes help in the transbilayer movement of lipids. So these kinds of enzymes that catalyzes the movement of the sphingolipids, phospholipids, phosphatidylserine and phosphatidylethanolamine are classified into three main categories, and they are known as flippase, floppase and scramblase. So what is done by flippase? So flippase basically catalyze, so it is basically catalyze the translocation of aminophospholipid, phosphatidylethanolamine, phosphatidylserine from outer leaflet to inner leaflet. And in this procedure, when this flippase catalyze the translocation of aminophospholipid, phosphatidylethanolamine and phosphatidylserine from outer leaflet to inner leaflet, they requires ATP molecule. So ATP is consumed in this process, and by this process, this flippase can actually transfer those aminophospholipid, phosphatidylethanolamine and phosphatidylserine from outer leaflet to inner leaflet. And these kinds of enzymes or flippases are examples of P-type ATPases. Okay, so this is the example of P-type ATPases or P-type pumps. So basically they cleaves the inorganic phosphate from ATP and ultimately produces energy, and by using those energy they can translocate those phospholipids from outer leaflet to inner leaflet. Another kind of enzymes are floppases. So floppases are basically ABC transporter family. They are belongs in ABC transporter family. So floppases comes under ABC transporter families, and basically floppases do, they transfer phospholipids from inner leaflet to outer leaflet. Okay, so floppases basically transfer the phospholipid from inner leaflet to the outer leaflet. Okay, so basically these are the examples of ABC transporter families or ABC transporter pumps. Another type of example or enzyme is the scramblases. Okay, so scramblases basically an enzyme that catalyze both directional movement, that means from outside to inside and from inside to outside, and by this procedure scramblases does not use any kind of ATP. So basically they does not use any kind of ATP and they acts along their concentration gradient. So they act along their concentration gradient. Okay, so these are the three types of enzymes that help the lipid molecules to move from one leaflet to another leaflet. One is the flippase, another one is the floppase, and third one is the scramblase. So flippase and floppase both enzymes requires ATP, whereas scramblase does not requires any kind of ATP. So flippases comes under P-type ATPases or P-type pumps. Floppases comes under ABC transporter family, and scramblases they does not require any kind of ATP molecule or for the energy or for the transfer of lipid molecules, because they acts as they are along with their concentration gradient. That means they moves molecules or phospholipids from higher concentration to lower concentration. So these are the transbilayer movement of lipids molecule. If you like that video, please hit the like button. Don't forget to subscribe, and thanks for watching.